Today I'd like to talk about a collaborative model. Today I'd like to talk about a collaborative model for bioinformatics education, combining biologically inspired bioinformatics with project-based learning. And this presentation is based on a collaboration between pine-biotech and the Loyola University of New Orleans, specifically a new class in bioinformatics that was organized by Dr. Kim Mix and other faculty for students that do not have a background in bioinformatics but were interested to join this independent study and learn about basics of bioinformatics in the context of project-based learning. So I want to start by pointing out that there is a dramatic gap between our ability to generate very precise high throughput data and then our ability to analyze, interpret, and apply that data. And this is a growing gap, particularly because more and more data is being generated. It's cheaper to generate data, and it's useful to generate data. However, the typical approach is that then once the data is generated, it is being taken to an analyst, somebody that sits at a bioinformatics department, does not know about how the data was generated, and doesn't know why it was generated. And this data is presented to them, explained to them, by somebody that speaks a completely different language. And that's why there was recently several studies that were talking about the gap in the skills, in the basic skills that are necessary for basic data stewardship, that they're only being taught at about only 25% of educational programs. So the challenge is to take this bioinformatics language, the language of data science, and the data about biology, so the language of biology, molecular biology, cell biology, and to bring those together into an integrated bioinformatics research experience and provide it for faculty and students in an integrated way, so that they're not a data science course and a computational course on programming in R, and then a separate lab experience about generating the data and RNA and DNA and the genes, but it's an integrated course. And the challenge really consists of two major parts, and so let me go through these challenges and then talk about how we could address these challenges. So first of all, to develop such an integrated course, you really need high quality professionals that are willing to lead and to service. It's not enough to have people that are willing to do bioinformatics research and take that research to a level of publications and grants and independent ideas, because ultimately they also need to go back and serve the other projects that might not be as interesting in terms of computer science, computational biology, and bioinformatics. The second one is really the costs of establishing and maintaining computational infrastructure. So a bioinformatics department really services a lot of projects. They need computational infrastructure that is dedicated for those purposes, but that infrastructure needs to be developed, maintained, and that is costly. And you can see that in terms of budget, the two main big portions of that typical bioinformatics department budget is staff and infrastructure, right? The other aspect, the other big portion of the costs and the challenges are really around training up and developing internal talent to be able to address the challenges. And so this is a lack of qualified trainers that have the experience in both computational and biological aspects. It's difficult to find people that have both sides of that paradigm. And then you have a lack of an easy to adopt curriculum. So there's a lot of books about bioinformatics, there's a lot of courses and technical resources out there, but there's not really a plug and play curriculum that is tested and customizable to address specific interests and needs in a integrated course. And then also there's an intimidating list of prerequisites. So if a student is potentially interested, he's faced with knowing statistics, and then knowing R, being proficient in R, and then knowing how to script in Linux, and all of these different prerequisites. And for a biologist, that's a major hurdle. A lot of times people tend to choose not to even take a course in bioinformatics because of these prerequisites. So how can we address these challenges and this need, and prepare something that is scalable, that is practical, and that really develops good skills for students? So first of all, it's learning by doing, right? So these are some principles that we came up with that became a part of our course that we are piloting now at University of Loyola. Number one, learn by doing. So dive into bioinformatics from the very first day. You start doing bioinformatics, taking data sets, analyzing data sets, interpreting data sets. Number two, online discussions. So it's very difficult to bring people together. It requires a lot of time, but people do spend a lot more time online today. And so we enabled this process of discussions to share experiences and issues with analysis, to be able to ask those questions of experienced bioinformaticians and review and reflect on their doing process. Then its ability to generalize. So not only some niche project that you focus on and you only do that project for 2 years. It's ability to learn skills in one set of projects and then apply them to other types of projects where you understand both the opportunities and the limitations of the approaches that you have been doing. And then finally, it is applying those skills that you have developed in a new way to understand real life situations, and that is to have a continuity. And after your project, after you've done a project, a test project, an educational project, to be able to launch your own research. And I think here bioinformatics provides a unique opportunity. It's a very different setting than, let's say, molecular biology, where you have to collect samples and prepare those samples and then do something. Really, there's a lot of data, a lot of resources out there that you can just tap into, and you can conduct your own research, because only a small portion of the data that today is available in repositories is fully analyzed. And then, of course, doing that all over again, right? So going back and taking on a different data type, learning about RNA-seq, learning about DNA-seq, learning about mass spec data, structural biology data, integration. So all of that process could continue. How do we do this technically? So first of all, a big resource that is allowing us to do this efficiently is the T-bioinfo platform. The platform that was designed for scientific research allows performing complex data processing and analysis pipeline without coding. And so here we are really lowering the bar for entry. People without any coding background can enter and try out very complex and computationally intensive pipelines, run them through, see what the outputs are, and understand how they might be useful. Not only that, the platform is visual, interactive. Every button that you see, it comes with a pop-up, and the pop-up both explains what the button does, the algorithm, and also provides more resources to learn about the algorithm, typically a publication, and a summary of what you will find it useful for. And the same interface that you see here is applied for many different data types. So you have all kinds of next generation sequencing data pipelines, mass spec pipelines, structural biology data, integration, and dedicated sections for virology and other unique sets of challenges. And so what's good about this is that you have a dedicated section that is compiling the right kinds of tools around an area or a data type or an application. For that data and for those analyses. Now, in parallel with this online platform, we also have a set of online courses, and it's an online learning environment that is all focused around bioinformatics. It has a number of courses, for example, Transcriptomics 1. So Transcriptomics 1 would go through how to take raw FASTQ files, next generation sequencing files, process those files, and get a table of expression. Transcriptomics 2 then picks it up from there, goes over a review of how that is done in a pipeline. You can do it. And then finally talks about differential gene expression and t-test statistics and what that means. And Transcriptomics 3, machine learning for transcriptomics data. How do you take the table of expression and work with the table of expression to classify samples or to know which features are most informative for classification, so feature selection, etc. And so all of these are centered around an individual, the student. The student sees the progress, can monitor their test results, can see how he compares to other participants in a course, and then of course we have an online chat, and so any questions or challenges he can ask the person, and the person is a trained bioinformatician that will answer it from an experience perspective. Now, these basic skills prepare students to take on projects. And here you see a number of example projects that we've prepared. These projects have curated data sets that were designed around specific skills, and they also expose the student to a real research environment. So not only do you have the data, you have the publication about the data, you need to be able to learn from that publication. And so, for example, when you take a project like this, modeling precision treatment of breast cancer using multiple types of cell lines and therapeutic agents, what you really get here is a data set that is fairly complex. It was taken from a number of other publications and then published as a meta-analysis. And so we offer a small portion of that as an educational data set and then provide the full project, and that project could be done independently by a student with their own research question behind it. Currently, we are doing workshops and we are preparing other projects and other courses that are published online. A lot of these resources are free. Some of them are paid. Some of them are done in collaboration with other university researchers and industry. And now we are giving these workshops and guest lectures, presenting the opportunity to implement this curriculum, design it in such a way that it fits the needs of different levels of students. We have over 2,000 students enrolled online. We presented the solution in a number of places, including Georgetown, NYU, a couple of universities in Nebraska, here in Louisiana at LSU, Tulane, University of New Orleans, and we have been selected to present at various startup competitions. At the same time, I want to mention that April 17 and 20, we are starting our online workshops. The first workshop is going to be at University of Nebraska Medical Center, and it's going to be jointly certified as a participation certificate. It's going to be a 3-hour workshop on next-generation data sequencing analysis, data analysis from NGS data, and it will touch on topics like processing data, quality control of the data, analyzing the transcriptome, analyzing the expression levels using machine learning methods, and then also looking at various biomedical applications of the data analysis. And the following workshop that we're going to have is going to be at University of Nebraska-Lincoln. All the same topics, but more catering to engineering students. Finally, I want to point out that we have this pilot and Dr. Kim Mix, who has been leading this pilot with over 12 students from various backgrounds and various departments, and she has an exciting program that combines various aspects of molecular biology and genetics and incorporating data science for those students as a way to prepare them for careers after they complete the course. And we are very excited about this project. It's still an ongoing pilot, but all the students have shown interest in continuing to learn about bioinformatics, continuing to learn bioinformatics, and after completing the course, they will actually present their independent projects to the faculty for evaluation and for feedback. So this is, in short, our approach to bioinformatics, our approach to incorporating bioinformatics into a biology curriculum. I think this is a very flexible way. It could also be adapted to the research interests of faculty so that faculty research and education go hand in hand. I'm constantly posting more information about these projects and others, and you can find pine-biotech on all the social media platforms, as well as you can look at some of these free courses that are available online. Just visit edu.t-bio.info. Thank you.